We're going to look at chapter 24 over the urinary system. The urinary system includes the kidneys, ureters, urinary bladder, and urethra. The kidneys are going to be the main organs of the urinary system because they not only carry out excretory functions like the other parts of the urinary system, they are also able to regulate total water volume and total solute concentration in water. They're also able to regulate extracellular fluid ion concentrations, as well as ensure long-term acid-base balance. And they're responsible for the removal of metabolic waste like urea, toxins, and drugs. The kidneys are able to do this because they contain nephrons. Nephrons are the structural and functional unit of the kidneys. Nephrons are essentially filters, and each kidney contains over 1 million nephrons. Other functions of the kidneys include the endocrine hormone secretion of renin and erythropoietin. Renin can regulate blood pressure, and erythropoietin regulates red blood cell production. We also see that the activation of vitamin D and the gluconeogenesis that occurs during prolonged fasting are kidney functions. The organs of the urinary system again are the kidneys, ureters, urinary bladder, and urethra. There are 2 kidneys, 2 ureters, 1 urinary bladder, and a urethra. The kidneys are the major excretory organs. Not only do they create filtrate, which eventually becomes urine, they send that urine from the kidneys to the bladder using the ureters. The ureters are transporting muscular tubes, and the ureters are going to enter or fuse with the urinary bladder at its posterior side. The urinary bladder is a temporary storage reservoir for urine, and it allows urine to leave via the urethra, and the urethra is a structure that transports urine out of the body. We can see here the 4 organs of the urinary system, the 2 kidneys below their adrenal glands, the ureters, which connect the kidneys to the urinary bladder, and the most inferior urethra. The vasculature of the kidney is vast, but what we're looking at here is some basic vasculature. We can see that the renal artery and the renal vein branch off of either the abdominal aorta or inferior vena cava, respectively. Because of the position of the abdominal aorta and the inferior vena cava, we also have differences in length for the renal arteries and veins to each kidney. For example, the left kidney has a longer renal vein than the right kidney because of the positioning of the inferior vena cava. The kidneys themselves are retroperitoneal because they sit behind the retroperitoneum. So they're in that superior lumbar region, and the right kidney will be a little bit lower than the left because it's crowded by the liver. They're below the adrenal glands, which sit atop each kidney, and they have a convex and concave surface. The concave medial surface is where we would find the renal hilum, or the helix, and the helix is going to be the location of where things enter or exit the kidney. In general, there's 3 large structures we can see enter or exit each kidney, and that is the renal artery, renal vein, and the ureter. Here is a position or image showing you the position of the kidneys on that posterior body wall. And so you can see they're kind of in that superior lumbar region, and they're going to sit really close to the dorsal wall of the abdominal pelvic cavity. If we're looking at the external anatomy of the kidney, we see that there are 3 layers of supportive tissue surrounding the kidney itself. Surrounding the renal cortex is the fibrous capsule, and it prevents the spread of infection to the kidney. Outside of the fibrous capsule is a fatty cushion called the perirenal fat capsule, and then there's the renal fascia that anchors the kidney in place against that dorsal body wall of the abdominal pelvic cavity. So the renal fascia is dense fibrous connective tissue, and it holds the kidneys in place. So here we can see those 3 layers. First, we can see the peritoneal cavity and how the kidneys sit behind it towards the posterior or the back of the abdominal pelvic cavity. So they're retroperitoneal. Each kidney has a renal artery and vein in this image, entering or exiting at the helix. We can see a light orange renal capsule, the fatty perirenal fat capsule, and then kind of a darker pink renal fascia. So those are the 3 layers we see as supportive tissues outside the kidney itself. If we look at the internal anatomy of the kidney, there's also 3 layers. That renal capsule was adjacent to or close to the renal cortex, which is the outer layer of the kidney. So this light pink here, That's going to be the cortex of the kidney. Then you have a middle layer, or the medulla. The medulla contains medullary pyramids as well as renal columns, which are these pieces in between each pyramid. The pyramids themselves have a base and then a point or apex, and the point or apex of each medullary pyramid is called a renal papilla. So the medullary pyramids and the renal columns are a part of the renal medulla, the second layer of the internal anatomy. And then the third layer of the internal anatomy for the kidney is the renal pelvis. The renal pelvis is a basin-like structure that is continuous with the ureter. So in order to create urine and have it leave the kidney, we would have to filter blood in the cortex using the nephron, and then it would travel from cortex to medulla and then to the pelvis. Other structures we see here are things like the minor calyces, found near the apex of each medullary pyramid. Minor calyces are funnels that lead to major calyces. Major calyces are the funnels created when many minor calyces join together, and then major calyces merge with the renal pelvis. So if we do this very simply and we just track the basic route of urine from the cortex to the pelvis, we would say that it leaves the nephron, then it travels through the collecting ducts of the medullary pyramids from base to renal papilla. From the renal papilla, the urine enters a minor calyx first, then a major calyx next. From the major calyx, it then moves into the renal pelvis, and then it'll flow into a ureter, where it then makes its way to the bladder and then finally leaves the body via the urethra. But what we saw was that filtrate from a nephron, that's fluid in the nephron, becomes urine as it travels from the cortex to the pelvis of the kidney. So just a quick review, there were 3 layers protecting or supporting the kidney, and that was the perirenal fat capsule, the fibrous renal capsule, and the renal fascia. Not in any particular order there. And then the 3 layers inside the kidney, or the tunics, or the 3 parts of the kidney, if we're looking internally, were the renal cortex on the outside, the middle renal medulla, and then the deep renal pelvis, which is continuous with the ureter. So we have in this PowerPoint more on the internal anatomy. So you can see here the definitions for renal cortex, the granular appearing superficial region, where you find a majority of the nephron. There's going to be structures of the nephron that proceed into the medulla, but most of the nephrons are in the renal cortex, especially the renal corpuscle. Then you have the renal medulla, made up of those medullary pyramids and their columns. And the columns are inward extensions of cortical tissue, but they are officially a part of the renal medulla. There's also the papilla, which was the point or tip, or maybe you can call it the apex of the medullary pyramids. And the medullary pyramids, again, are a part of the medulla, and they are going to contain collecting ducts. The collecting ducts actually give the medullary pyramids a striped appearance, which is what we can see over here on the right. A lobe of a kidney actually includes the medullary pyramids and about half of the renal column that's adjacent to it on both of its lateral sides. And then last but not least, there's the renal pelvis, which is a funnel-shaped tube, again, continuous with the ureter. The renal pelvis is continuous with the ureter. From the renal papilla, urine would enter first into a minor calyx, then a major calyx. And plural for that would be minor calyces and major calyces. So again, if we just track that urine flow using all this terminology we're learning, it would leave the renal pyramid via the renal papilla, enter into a minor calyx, then a major calyx, next the renal pelvis, and then flow into the ureter. So just a review of structures. The 3 regions of the kidney are the renal cortex, renal medulla, and renal pelvis. The renal cortex mainly contains nephrons. Only parts of them reach into the renal medulla. The renal medulla is made of medullary pyramids as well as renal columns, We would define lobes of the kidney as a medullary pyramid plus half of its adjacent renal columns. And the renal pelvis is going to accept urine from the minor then major calyces, and it's going to be continuous with the ureter. Let's just talk briefly about some homeostatic imbalances. There's pyelitis, which is infection of the renal pelvis and the calyces. And then there's pyelonephritis, which is infection of or inflammation of the entire kidney. Now, normally these can be successfully treated with antibiotics. However, because the kidney and its renal pelvis and other of these cup-like or basin-like funnels are continuous with the ureter, we can have infections that originate either in the urethra or in the bladder, go up the ureter and infect the kidney. So that could be the cause or the initial location of the bacteria that causes pyelitis or pyelonephritis. So let's just review real quick. Our organs of the urinary system are the kidneys, the ureters, the bladder, and the urethra. That list goes superior to inferior. We can find the organs of the urinary system in the abdominal pelvic cavity. You have 2 kidneys, 2 ureters, 1 urinary bladder, and 1 urethra. The basic vasculature that we've looked at thus far for the urinary system includes the renal arteries that bring blood to the kidneys, and then the renal veins which drain blood from the kidneys. We also saw that the positioning of the inferior vena cava and the abdominal aorta lead to some slight structural differences in the length of the renal artery and vein for the kidneys. Also, because of the liver, the right kidney is going to be crowded out a little bit in that abdominal pelvic cavity, so it's going to be a little lower than the left. So we're going to look more at that blood and nerve supply now for the kidneys. And we know the kidneys are great filters. They contain those nephrons, so they can cleanse the blood. That's what the nephrons do, is they filter stuff from the blood, and then the rest of the nephron is going to basically adjust that filtrate and take back what they want. And in the blood, if it figures out that it has too much sodium or potassium or there's waste products, they'll secrete that to the renal tubule system. So there's a couple of parts of nephron we'll look at, but kidneys are going to cleanse the blood. And it's able to adjust that composition. But because they're using the blood as the primary way that they create this filtrate is what's in the nephron and eventually becomes urine, they have a very rich blood supply. You see here it says that the renal arteries deliver about 1/4 or about 1,200 milliliters of the cardiac output. So that's what your heart is pumping out from each ventricle per minute. It delivers about 1/4 of that to the kidneys. So the kidneys are constantly just filtering your blood supply. So that arterial flow into the kidney and the venous flow out of the kidney have very similar paths. There's a couple of differences, but pretty much we just changed the name of the arteries to veins when the blood flows out of the kidney, and we'll look at that pathway. But after talking about this, you should definitely be able to answer any question that says, are the kidneys vascularized? As true. They have a very rich blood supply, a highly vascularized network of veins and arteries. But they're also innervated. They have a nerve supply. They have a nerve supply that mainly uses sympathetic fibers, and they are called the renal plexus. So here is the basic vascularization of the kidney. I say basic because once you get the renal artery part, you have the renal vein part. I swear you have it down. Just take some practice. So let's try it out. So I'm going to try to use my pen. So let's see if we can follow the blood supply in the kidney. So we're going to start in the renal artery. And from the renal artery, blood will enter the helix. And then we have some branches. These 2 branches are segmental arteries. And the segmental arteries are then going to branch and go up in between our pyramids. So we start in the renal artery. The first branch, we have branches into the segmental arteries. And then the next branch, which goes up in between the pyramids, that is going to be called the interlobar arteries. So now it may be apparent that it's important to know what a lobe for a kidney is. Because if you know that the renal pyramid and part of the renal column is a lobe, and now we have this artery going between the lobes, it makes sense that we're calling it an interlobar or between the lobe artery. So renal artery, segmental artery, interlobar artery, and now look at this artery that arcs over the base of the medullary pyramid. So it's going to take a right, or it could take a left, and it's going to make an arc over the base of the medullary pyramid. And that artery is called the arcuate artery. And we have a couple of different arcuate arteries. If you look at the base of all these medullary pyramids, they have arcuate arteries. And then from the arcuate artery, we're going to have a branch that goes off into the cortex. So that artery that goes into the cortex or radiate out into the cortex region is actually called the cortical radiate artery. It's also known as the interlobular artery, but to me that sounds a lot like interlobar, so I do like that your book, your lecture book, uses cortical radiate instead. Just be aware that interlobular is actually a term used for the cortical radiate artery and the vein. Now, we haven't looked at the microscopic structure that's in the cortex, so there's more blood supply. We don't just immediately back out of the kidney now and go back towards the renal vein. So I'm going to list for you the vasculature that you can't see in this image, and eventually we'll look at that in more detail. So from the cortical radiate artery, we would have a branch that would go to the nephron called the afferent arteriole. That's afferent with an A. The afferent arteriole leads to the glomerulus. The glomerulus is a capillary bed. That capillary bed is the first capillary bed we've talked about for the kidney, and it is drained by another arteriole. The arteriole that drains the glomerulus is called the efferent arteriole. So efferent, that's with an E. The efferent arteriole goes into or becomes the peritubular capillaries or the vasa recta, depending on the type of nephron you're looking at. There's two types, but the peritubular capillaries and vasa recta, those are the second capillary bed. So I'll stop right there for just a second. That's interesting for a tissue. Usually we have one capillary bed, like pulmonary capillaries, for example, and we have an arteriole that lets blood flow into the capillary bed, but it's a venule that drains the capillary bed. So this is interesting. The kidney has two capillary beds and two arterioles. So its vasculature is very unique. And so make sure you're aware of how unique this vasculature is. Not only do we have two arterioles, one having blood flow to the first capillary bed, one draining it, we have another capillary bed as well that's going to function in both reabsorption and secretion of the tubular system of the nephron. Now, to continue the vascularization, after we leave the peritubular capillaries, or vasa recta, blood will then flow into the cortical radiate vein. So now we're back to what we can see on this image. So here in blue, let me see if we can change the color. Okay. So here in blue, we'll use this one. Or maybe not. Okay. So here in blue we have, or red, we'll just stick with red. We have the cortical radiate vein. From the cortical radiate vein, we have a branch, which is this blue right here. That's the arcuate vein. So blood's now backing out of the kidney. And then blood will go from the arcuate vein down through the interlobar vein here in blue. And then it'll go right back into the renal vein. So we sort of went out the same way we came in, but we skipped the segmental vein. They don't exist. So I'm just going to review the way we went in and the way we went out and see if you can pick up on some of the changes. All right, so blood enters the kidney via the renal artery. The renal artery then branches into the segmental arteries. The segmental arteries branch into the interlobar arteries. The interlobar arteries then branch into the arcuate arteries. The arcuate arteries then branch to the cortex and are called the cortical radiate arteries. The cortical radiate arteries lead to the afferent arteriole, which leads to the glomerulus. The glomerulus is drained by the efferent arteriole, which leads to the peritubular capillaries or vasa recta. The peritubular capillaries or vasa recta then lead to the cortical radiate vein. The cortical radiate vein then leads to the arcuate vein. The arcuate vein flows into the interlobar vein, and the interlobar veins flow into the renal vein. So I basically went towards the cortex and then back, and it pretty much went towards and back in the kidney, except for the segmental vein the same way. So there's a little exception there that segmental vein doesn't exist. It goes straight from interlobar vein to renal vein. Now, I wanted to look at the actual anatomy of the kidney first before I just start throwing out terms like segmental artery, afferent arteriole, or cortical radiate vein. So here's a pathway that might be more helpful now that we have a general idea of where those structures are. So let me see if I can just kind of summarize where these structures are. I'll try to write them. We saw, for example, the renal artery and the renal vein at the helix. So we're going to enter via the renal vein, and one of the last structures we're going to exit from is we're going to enter via the renal artery. One of the last structures we're going to exit is via the renal vein. Now, the abdominal aorta is feeding the renal artery, and from the renal vein, we're going to see blood enter into the inferior vena cava. So those are structures we saw when we were looking at the basic vasculature for the kidney. All right, so we found those at the helix. The renal artery leads to the segmental arteries. So those are the first branches from the renal artery located inside the kidney. So in humans, they're located inside the kidney, those segmental arteries. And we saw them about in the, we'll say like the renal pelvis region. Then the segmental arteries lead to the interlobar arteries. And their name is great because they're located between kidney lobes. So they're located between those medullary pyramids and the renal columns. Next, we have the arcuate arteries, also a great name because they arc over, or they kind of take a right turn or left turn, depending on which one you're looking at. They arc over the base of the medullary pyramids. And then cortical radiate artery tells you what it does. It radiates out into the cortex of the kidney. And before we look in this kind of dotted box, let's talk about the way we just entered the kidney. So cortical radiate artery is the last structure we were in before we move into the microscopic structures, which are in this dotted box. Notice that after we leave the dotted box, we're almost going to be moving out of the kidney the same way that blood flew in. All right, so before the microscopic structures, we were in a cortical radiate artery. After the microscopic structures, blood's going to flow into a cortical radiate vein. After the cortical radiate vein, blood will flow into an arcuate vein. After the arcuate vein, blood will flow into an interlobar vein. And remember, we skipped the segmental because there's no segmental vein, but after the interlobar vein, blood's going to flow into a renal vessel. Here it's the renal vein instead of the renal artery. So slightly similar there. We'll do a dotted line. So that means if you're studying and you're trying to really get this vasculature down, you just have to get this part down. And then remember that we have no segmental vein, and then it's pretty much backwards. And then the only other part you have to study is this part right here, which is microscopic. So we didn't see that on the image in the last slide. And that is the two arterioles and the two capillary beds. So at the very least, you know that the microscopic structures have to have those four things. And the afferent like arrive, that's going to bring blood to the first capillary bed. The efferent like exit, it's going to drain the first capillary bed. And then once we drain the first capillary bed, we have to move into the second. So remember, depending on the type of nephron we're talking about, could be paratubular or vasorecta for that second capillary bed. So we'll get to that, but those are your two options for second capillary bed. So I'm just going to list these so you've heard the terms probably for a fifth time now, but this is the vasculature of the kidney. Aorta, renal artery, segmental artery, interlobar artery, arcuate artery, cortical radiate artery, also known as an interlobular artery. Then we move into the afferent arteriole, glomerulus, then the efferent arteriole, then paratubular capillaries or vasa recta, then cortical radiate vein, also known as the interlobular vein, then arcuate vein, then interlobar vein, finally renal vein, then inferior vena cava. So just practice that vasculature. And don't forget what makes the blood vessels or the vasculature for the kidney unique. There are 2 capillary beds and 2 arterioles. So now let's look at those nephrons. Let's look at our structural and functional units of the kidney. And there's about 1 million per kidney, and there's 2 main parts. So the 2 main parts of the nephron are the renal corpuscle and the renal tubule. And we're going to look at those a lot closer. So the renal corpuscle is made up of the glomerulus and the glomerular capsule, also known as Bowman's capsule. So let's just take a step away from all the information on this slide. We're looking at a nephron. A nephron is a microscopic structure. Most of it is located in the renal cortex, especially the renal corpuscle of the nephron. So we're looking at that one part of the nephron that's always located in the cortex, the renal corpuscle. Okay. The renal corpuscle is made up of some stuff. It's made up of that first capillary bed, the glomerulus, and a capsule that surrounds the first capillary bed. That capsule is named after the glomerulus, glomerular capsule, but also has another name, Bowman's capsule. So glomerulus is a capillary bed. It's fenestrated, that means it has pores, so it's highly porous, and it allows for glomerular filtration, which is just filtration with glomerulus in front of it because that's where it occurs. So glomerular filtration occurs where the capsule and the glomerulus meet. So this is allowing filtrate formation. Filtrate is the fluid that enters the nephron. Filtrate eventually becomes urine. The capsule itself does just surround that glomerulus, so it's cup-shaped. There's a layer of the capsule that touches the glomerulus, and then there's a layer of the capsule that doesn't, and there's a space between them. And the filtrate first leaves the glomerulus and enters this hollow part of the glomerular capsule. So let's look at the nephron. Now thus far, we're focusing right here, just the renal corpuscle. So we're looking at the glomerulus, which is this kind of wiry looking thing, inside a capsule. That's what we're looking at. We're looking at the glomerulus and its capsule. Together they make up our renal corpuscle. And we know that the renal corpuscle, made up of those 2 things, is always found in the renal cortex, which is the superficial region under the renal capsule. Now, next we're going to look a little closer at that renal corpuscle, and then we'll move into what is called the renal tubule system. And we'll see that there's also going to be parts to that renal tubule system. And if you're looking at the right and you're seeing all of these cell types, we're going to get to that too. All right, so the renal corpuscle consists of that glomerulus, that's our capillary bed. We've pretty much covered most of the stuff that has to do with it. It's a capillary bed, it has pores. We know pores are called fenestrations, but we're kind of more interested in this capsule. We haven't dealt with a capsule around a capillary bed like this, not yet. And so we said it had 2 layers. The layer that touches the glomerulus is the visceral layer. The layer that doesn't touch the glomerulus is the parietal layer. And that parietal layer is going to be made up of pretty much simple squamous epithelium, really thin cells. But the visceral layer, it has special cells. These cells have branching epithelial podocytes. So podocytes, podo is a prefix that means feet. These are kind of like feet cells. They have branching extensions that look like feet, and they touch the fused basement membrane between the endothelium of the capillary bed and these cells. So the visceral layer is made up of branching epithelial podocytes or foot cells. Not really foot cells, but it helps to remember them. Now, these foot cells use their foot processes to cling to the basement membrane, which is the basal layer of the glomerulus, or the endothelial cells of the glomerulus. And they don't cling in a continuous layer. There's spaces because of these foot processes. Like one foot hits one area, one foot hits another area, and there's a space between the feet. Like your own feet, if you're just standing. Those spaces are called filtration slits. And the filtration slits between the foot processes are spaces that allow filtrate to flow from the pores of the capillary bed, your glomerulus, into the hollow space of Bowman's capsule, which we also know as the glomerular capsule. So let's actually look at these structures. So here's the parietal layer. It's not really functioning in filtration. It's just there to help kind of complete the cup, which is our capsule. So simple squamous epithelium works just fine. Now here's our foot cell. Looks a little creepy. Kind of looks like it has one eye. But we can see all of these foot processes. And it's not forming a continuous barrier. Instead, we have spaces. So here's a space, here's another space. You kind of get the idea. We have filtration slits. And over here, we have holes in the endothelium of the glomerulus, which is a capillary. So the glomerulus is just a blood vessel. It's a capillary bed, so they're really small blood vessels. And it's a fenestrated capillary bed, so there's holes in the epithelium. If you remember back to the cardiovascular chapters, capillaries are made up of one layer of tunica intima, or simple squamous epithelium. So we have really thin cells with holes in them, and that's what we're seeing here. Fenestrated endothelium of the glomerulus. Fancy way to say one layer of simple squamous epithelium with holes in it. We have here in blue the fused basement membrane. And that fused basement membrane is just going to be between the podocyte and the capillary cells. And it's just going to help hold them together. But notice we have holes in the capillary bed. We have slits between the podocyte foot processes. So if fluid wants to leave the capillary bed and it fits through the pore and it fits through the filtration slit, then it gets to move into the capsule. So what we're seeing here is a filtration membrane. We're seeing an array of cell types arranged in a certain order, and that order, that structural arrangement, will allow stuff to enter the glomerular capsule or Bowman's capsule as long as it fits. So really small nutrients, things like sodium ions, potassium ions, urea, these small things can move into that capsular space. So if we just want to list real quick the three things that make up the filtration membrane, these would be the three things. The foot processes of podocytes, the fenestrated endothelium of the glomerulus, and the fused basement membrane. Those make up our filtration membrane. All right, so now we've pretty much looked at the renal corpuscle. Next, we're going to move on and look at the renal tubule system. The renal tubule system consists of three parts. The proximal convoluted tubule, which is this windy piece right here. It's a tube, so tubule makes sense. It's proximal because it's really close to the renal corpuscle. We can compare or contrast that with our distal convoluted tubule, which is convoluted and bendy. It's a tube, so tubule makes sense. And distal because it's farther away from the renal corpuscle than the proximal tubule. So proximal and distal here are just giving you kind of an orientation, which one's close to the renal corpuscle, which one's not. Then in between the proximal and distal convoluted tubule is our loop of Henle, or sometimes just called the nephron loop. Now we have a descending limb and an ascending limb of the nephron loop. And then down here at the bottom, we have a hairpin turn. So hairpin turn. That's supposed to be an H. We'll just move on. All right, so renal tubule system, three parts: proximal convoluted tubule, nephron loop, and distal convoluted tubule. You can abbreviate the proximal convoluted tubule as the PCT, the distal convoluted tubule as the DCT, and then know that the nephron loop is also known as the loop of Henle. Henle being spelled Henle with a capital H. The PCT has a lot of cuboidal cells. These cuboidal cells have a brush border or microvilli. And remember, like with the digestive system, the microvilli were able to increase the surface area. So they're increasing the surface area for absorption. Now, these cells themselves with their microvilli are going to function in some type of absorption. This function is going to be in reabsorption. Now, it's called reabsorption because this will be like the second time that nutrients are being absorbed. And the nutrients originally were absorbed through the enteric epithelial cells of the duodenum or other parts of the small intestine. So now they can be reabsorbed or absorbed for a second time in this proximal convoluted tubule. And most nutrients will be reabsorbed back into the blood during this process. So tubular reabsorption primarily occurs in the proximal convoluted tubule. This includes the reabsorption of water, ions, and nutrients that originally entered Bowman's capsule via glomerular filtration. So make sure you know that glomerular filtration happens at the filtration membrane of the renal corpuscle, but it's not very selective. Anything that fits through the filtration membrane goes into Bowman's capsule, which then has its fluid called filtrate flow into the proximal convoluted tubule. So what happens is the blood wants to reclaim stuff that was taken from it because it wants some of its sodium ions back, it wants nutrients like glucose and other things back. So it'll reabsorb the things. It'll reabsorb from the renal tubule system back into the blood. Now, secretion also occurs in the proximal convoluted tubule, and secretion is the opposite of tubular reabsorption. Tubular secretion is when waste products or ion excess or other things in excess leave the blood and move back to the tube. So, tubular reabsorption is when things go from the tube to the blood. Tubular secretion is when things go from the blood to the tube. We find that both the proximal and distal convoluted tubules are confined or located in the renal cortex. So let's look at those cells of the proximal convoluted tubule with their microvilli, which are located on their apical layer. They also contain plenty of mitochondria for active transport of substances from the tube back into the blood. Next, we're going to look at the nephron loop, which has descending and ascending limbs. It's going to be connected on one end to the PCT, the other end to the DCT. And its distal or descending limb has a thin part and a thick part. And the thick ascending limb will have cuboidal to columnar cells, but we're mainly going to look at the thin part of the limb or the nephron loop, and it's going to mainly have simple squamous epithelium. So that's what we're seeing here. Just general cells, pretty thin, flat cells. They do have the regular organelles. They have a nucleus, they have endoplasmic reticulum, and mitochondria. Whereas the cells of the distal convoluted tubule are going to go back to being cuboidal. So we saw cuboidal cells in the PCT, and cuboidal cells now are in the DCT. But a main difference here is we don't really see that many microvilli on the cuboidal cells of the DCT. Instead of functioning in reabsorption, which is the primary function of the PCT or proximal convoluted tubule, the distal convoluted tubule functions more in secretion, more in tubular secretion, or really getting waste products from the blood back into this renal tubule. And again, it's confined to the cortex like the PCT was. Here's its cuboidal cells, not as many microvilli, less involved in that reabsorption. And then last but not least, we will look at the collecting ducts. So there's two cell types in the collecting ducts. We haven't really mentioned them yet. I think they were mentioned once at the beginning of this PowerPoint, and we located them or gave them the location of running through the medullary pyramids. And they give the medullary pyramids a striped appearance. These collecting ducts are not a part of the nephron, but they do merge with multiple distal convoluting tubules of nearby nephrons. So after filtrate is made, it runs through the renal tubule system. Then when it leaves the nephron, it next enters into collecting ducts. So this is the structure we're looking at after the nephron. It has principal and intercalated cells, and the principal cells don't really have microvilli, whereas the intercalated cells have a lot of microvilli. And we see that the principal cells are involved in regulating water and sodium balance, and the intercalated cells are involved in regulating acid-base balance. So intercalated cells regulating acid-base balance, tons of microvilli. Principal cells more involved in kind of sodium and water regulation. So again, the collecting ducts are receiving filtrate from many nephrons. The collecting ducts themselves run through our medullary pyramids. They give the medullary pyramids their striped appearance. The many collecting ducts fuse together at the renal papilla of the medullary pyramids, which then drain to the minor calyces. So what we've done is we've worked our way from the cortex and through the renal medulla. We have the nephron creating filtrate, and then we have it also giving that filtrate once it's made and gone through some processing to the collecting duct. We have the collecting ducts running through the renal pyramids, and then we have them running through the pyramids from base to papilla, and then the papilla are going to allow fluid or filtrate from the collecting duct enter into the minor calyces. The minor calyces are accepting what is essentially now urine from the renal papilla. And remember, the minor calyces will then empty into major calyces, which are continuous with the renal pelvis, which is continuous with the ureter. And we're going to stop there for this part of the PowerPoint. Next time, we're going to look at the different classes of nephrons, and you can see here we have two types to get into.